Hey all, this is the PS Podcast, and I'm your host Chaitanya. The following podcast was recorded in September 2019, but the conversation has remained fresh with me ever since. For today, our guest is Dr. Annie Hassan. Dr. Annie is a pioneer in the field of genetics for India. She holds a PhD in genetics from Osmania University and has over 32 years of research experience. Her areas of interest include molecular pathology and cancer, neurodegeneration, diabetes related complication, and pharmacogenomic based medicine. Dr. Aini has spent 8 years at Wellington Medical School, New Zealand, and has over 100 international publications to her name. She is currently the senior consultant and the HOD of the Department of Genetics and Molecular Medicine at Kamineni Hospital, Hyderabad. She is involved in research, training, diagnostics, and counseling. She is also the founder coordinator for the first full-time program in genetic counseling for India. Our interaction with her was quite pleasant and informative at the same time. But because of her busy schedule, we had to plan the meeting at her office, which was unfortunately a victim of some background noise. The sound quality, though bearable, may be subpar, but as promised before, the content of the exchange will far overshadow any hindrances. We have also put subtitles on YouTube that can help you follow through the whole conversation. I met Dr. Aini at IIT Hyderabad during one of her lectures. She seemed extremely articulate and fascinated about her own subject. I thought she would be a wonderful speaker for this platform, and she was. I must concede that before this meeting, I had just completed the book Gene and Intimate History by Siddharth Mukherjee, so I was terribly impatient to have this conversation with her. By the way, if anybody is wondering, the book is absolutely compelling, and anyone interested even a little in our microscopic biological messengers should pick it up today. We must warn the listeners that the language in this exchange can alienate you, as it is straight out of the genetic domain. But we try to address all the terms instantaneously whenever possible. In any case, we do encourage you to pause the audio whenever any terminology confuses you and look up the meaning online. It will only enhance the experience. The conversation starts off with us getting to know about how Dr. Aini got into genetics. We then stride along and she explains to us what a gene and a genome is. We talk about how much awareness there is regarding the scale at which genetics can affect our daily lives. We talk about the twin genomic research and how that has contributed to the studies in genetic behavior. She gives us a glimpse into what targeted gene therapy is and how it works. There is a small detour that talks about the thrifty gene hypothesis and why the sickle cell trait had its benefits for the regions endemic with malaria. We lightly touch up on the technologies of CRISPR and Cas9 and its application in India. We also discuss its role in rare diseases and germline cells. I ask her a question about whether the matter of genetic manipulation will become a central concern in the future and the dire need of genetic counselors. The conversation ends with talking about some sci-fi scenarios and finally about gender identity. The talk was fascinating to us as it shed light on a topic that is going to dominate conversation in future. If you find it interesting too, please do not forget to like and subscribe our channel. Also, press the little bell icon so that you can be informed whenever we drop a new video. Now getting to the topic at hand, we present to you Dr. Aini Hassan.
So I wanted to know how were you inclined towards biology or studying the medical field? Was it an inherent wish of yours or someone from your family maybe?
No, see, genetics, I would say, not so much of biology. I was always interested in genetics. When I was doing my 11th, 12th, we had a chapter called Heredity and Variation. And I was really taken up by that because I was standing at the bus stop and a person cycling down stopped and came up to me and said, "Are you Mr. Turavel Hassan's daughter?" I said, "Yes." "How do you know?" He said, "Your mannerisms, your voice, your face looks exactly like him. And you must know that when you see a school coming out and standing at the bus stop, even it's difficult to find your own child." Okay. So this was fascinating. I said, "Now this is genetics, correct? That how is it that I'm like him, but I'm different too?" So that became. I was misled into thinking that you have to do medicine to do genetics.
Yeah, because even I was thinking that way, like as it is the other way around. Like first you go into the field, then you specialize into something.
Right. So I wanted to do genetics. So that's why I got into those medical exams. But luckily for me, while writing those medical entrance exams, I went to All India Institute, I went to Aligarh University, I went to Osmania Medical College, and I realized that clinicians do not have a clue about genetics.
What year was this?
This was in 1979-80. Okay. So, but I learned that there is an Institute of Genetics which is really started at Begumpet. Went and saw there, and there was no clinician there. We were all in genetics. So that's how I deferred medicine and went into graduation, masters, PhD, then came from that side into health sciences, human role of genetics in health diseases. I learned molecular biology I got a Department of Biotechnology Fellowship to work in CCMB, and then I did with human genetics. I did my PhD in human genetics, and after going as a scientist abroad, I realized that their genetics is more in the clinical areas. Your genetics is more for research. And this again you must have felt in IIT also. All those people know genetics, but they don't know the connection with the application of genetics. So then when I came back from New Zealand, I was there for 8 years working in the Wellington Medical School. From there we went to the US, Stanford and all. I realized that most of the applied genetics is in the medical school or in hospital. Basic research is different. And so I came back. I had an offer to go back. The choice was either I go back to CCME or these institutes where I get a cushy job and get on with my research, or try to take genetics to the hospital setting and patients, and also to educate the clinicians. And then I started working at Mahavir Hospital. So nobody understood what it was, so I never got paid for that initially. Then Kamineni Hospital, they said that they had a real vision about it. They said, "We know that genetics is going to come and it's the future, but we don't know what to expect. So could you tell us how to go about it?" And they asked me, and I gave them a 3-stage developmental plan that initially we could do chromosomes, then we could do. They agreed, and then from 2001 I set up that. Then I set up genetic testing in Vasvi Hospital, Mahavir Hospital, and you will not believe it, Institute of Genetics took my trained people to set up diagnostics for patients there later on. And also it helped with Handana Tambro. I changed the way, I digitized it, put in cameras, because before it was, taking pictures, cutting karyotypes by hand and all that. So then now we got images on computer. So people got more interested in learning that. Then I realized that there's more. Once we have a setup, we have to educate the clinicians to send you the patients. So I would go to pediatrics, gynecology. Yes, right. Then, but now I sit in my room and they send the cases and come in, so I don't have to worry.
That means you have been successful in what you tried to do.
See, my only success has been that from the IMR to the ward boy to the clinician to the MD know me as the genetics madam, so that's fine. So I've got genetics into the clinical setting.
And you've done it on quite a big scale, I can see that.
No, I don't know. I would like to do it on a larger scale.
See, of course, the domino effect will take its place because you started it. At least if I go today to my hometown and talk to them about genetics, I'm sure they would be dumbfounded. But here, if you tell someone, yes, ma'am ne bataya tha and this is what is happening.
So that's where, and now my students are there in Apollo, in KIMS, in Virinchi, so they have set up LV Prasad. So now that's setting up. Now LV Prasad, they also want a similar genetic unit in Vizag Center, in Orissa Center. So that's how it will cascade effect. Yeah. So I've answered your question. That my interest was genetics and not...
What I took from that is because as we are growing up, now we're married and we're growing, it seems more and more like one person can make a very big difference. Because that happens in your daily life. But hearing your story, just because you never deviated from your path and now it is giving you fruition, right? So it's very nice to hear. Even the people who are listening might, because a lot of times we take decisions that we don't want to take just because the time or the situation demands it.
So there was disappointment because my parents felt disappointed when I didn't want to do medicine, because that's a middle class ambition, correct? That the child should be a doctor or engineer or something. So they were disappointed, not anymore. But what I'm saying, at that point in time, they said, my father said, why don't you do medicine? At least exam to likho. My God, if I pass that exam, I'm stuck for life. I have to do genetics. So that, and now, funnily, I'm part of two medical schools where I teach postgraduate genetics to doctors. So that's serendipity or whatever I should say, that you become a teacher for a doctor without technically being a clinician.
And one more thing, when I attended your talk at IIT, it was, I'm being very honest here, I have never seen so many people in one talk, attending a talk. Yes, lectures they do attend, but a guest talk, so many people gathering up. I've seen maximum 20-30 people in one classroom. But that day, you must also remember, it was full.
Yeah, it was. But then that has nothing to do with me. That must be the organizer's credit. It was
a mail that is sent to the whole campus. We receive it, other departments also receive it. But I'm sure the subject itself is growing. So
Absolutely.
People are...
So now genetics has come into mainstream. And that is not my doing. It is the success of the Human Genome Project. Correct? Because people now know about it. People know that genetics is impacting everyday clinical practice. So, technology has advanced. And also now, all this social media, like your podcast, people will know that genetics initially was confined to pediatrics. People thought you were born with a genetic disorder. They are right, you are born with a genetic disorder, but the symptoms may appear at any stage in life. They can appear for thalassemia at the age of 1 year, for autism at the age of 2 to 3 years, at Alzheimer's in 60, 70 years, breast cancer at 30, 40 years. You are born with the gene, but the expression. So now the genetics is just exploding and getting into every...
Also, I might be wrong, but I've also read that the triggering of the expression might be because of some environmental cause also at some point of time. So you might have that gene and live your whole life without it affecting you, but in some places it might trigger. And
the most common example for this is the lactose intolerance. If you see, all mammals drink, as babies they drink milk. After that they don't. Correct? Whether it is a tiger, a lion, elephant, all the babies start their life with milk. It's only humans who continue to drink milk with the other food. So the gene which is for lactose digestion, it becomes less efficiently functional. And that is why certain triggers or certain things with the age of onset suppress that activity totally, and then you become lactose intolerant in later life.
Yeah. In fact, I read that most of the humans were lactose intolerant earlier, and once we started the agricultural revolution came.
Domesticated those animals which gave us milk, and we thought, yes.
So, to a lay person, how would you describe what a gene or a genome is, and how does it work?
Yeah. The gene I always describe as a unit which is given to you from your parents, and that affects a particular aspect of your physique. It can be your skin color, your curly hair, the color of your eyes, or getting diabetes or not. So something which is structural or functional. So it's a bit of DNA. DNA I never translate, but it's a molecule, it's a substance, it's something which is responsible for keeping details about your gene, what is happening, what's not happening. So DNA and gene, we don't need to translate it, but we just need to indicate it, that you get everything you get in two forms, one from your father, one from your mother. Sometimes one of this may get affected, so I show it with pictures or show it with just by fingers, that one is affected, one is normal. If both need to be affected to get some diseases, one needs to be affected to get some diseases. These changes can occur through environmental pollution, smoking, alcohol, drugs, whatever. So gene is, I always say, it's a unit which is being carried out from generation to generation, which gives you a particular characteristic. And it doesn't have to be good or bad.
I read this somewhere. So most of my knowledge that I'm producing here, I'm telling about, is coming from just one book, that is Gene from Siddhartha Mukherjee. I already informed you of that.
His family had psychobehavioral, neurobehavioral problems and psychiatric.
And you also mentioned this in your talk, that there is always some personal thing going on whenever somebody makes a leap in some frontier. So ma'am, I'll just explain to you what I have understood, what DNA and RNA do, and can you just point out if I'm wrong? So from what I know, DNA supposedly carries the information, and RNA is a copy, is a one-sided copy of a DNA, which detaches itself from DNA, goes into a ribosome. A ribosome studies it and then takes in amino acids and creates proteins out of it.
Yeah, this is what we call as the central dogma of molecular biology, that every living thing has to have nucleic acids, because DNA is the only living molecule of the millions and trillions of molecules in the world. It's a chemical molecule.
Other than that, it is just molecular interaction.
Just. So now the DNA has this ability of self-replicating, which is one of the characteristics of living things. So that's why DNA is very important. But this DNA is usually, during evolution, has been encapsulated in the nucleus. So the information which is there in the DNA has to go into, it is like DNA is the architect's plan, and now it has to be executed to become a house or a building. So the RNA takes this message so that it can be converted to bricks and cement and all to make the structural thing. So RNA takes the message from one of its side, and this message goes to the ribosomes, which act as the factory for making proteins. So ribosome doesn't interpret it. Ribosome just holds the links of the thread of RNA, and there is a separate machinery which brings in the amino acids based on the, like all languages, the language of DNA has 4 alphabets, A, T, G and C, and the language of genetics has words which have only 3 letters. English language has even is a word, TO2 is a word, TWO2 is a word. So it can have words which are single alphabet or multiple alphabet. But the language of DNA has only 3 alphabet words. So it can be TTT, ATG, CCC, like that. So various permutation combinations of these 4 give you 64 words. So it's fascinating that all living things are made with these 64 words. So from these 64 words, you have one word which says start. Now you have to start. So that is AUG. That starts it, which codes for one amino acid called methionine. So all of our proteins, whether they are structural, which is from the skin color melanin protein, or the insulin, it has to start with methionine, and then it keeps adding different amino acids and makes a unique protein. And these unique proteins all end when some nonsense words come, which are 3 nonsense words, which we call them stop codons. AUG is the start codon, and there are 3 nonsense words which say now stop. The amino acids keep adding, and then that's the end of the gene. So there's a start and an end. Remaining DNA does not code for much. So if you see all that human genome got trillions and billions of nucleotide sequence, but only 1% of it gives us meaningful proteins.
So am I right to say that those are introns?
They are introns as well as interspersing sequences. Introns are in between the exons of the same gene, but interspersing sequences are large DNA segments which don't code, but they do have function. We are realizing it now. Yeah. One of the functions is that they regulate the genes which are around there.
Okay, I'll move on to the next question now. So I'll just read a small paragraph. This paragraph is again from the same book, and I would like your comments on that. So there was this study by Dr. Bouchard, I don't know if I'm pronouncing it correctly, where he took pairs of twins, and especially those cases where they have been separated at birth. And then he called them for the study, and then he studied. Of course, there were a lot of variances in them, a lot of differences, but I'm just paraphrasing one paragraph from his study. Yet in Minnesota, Bouchard's staff was repeatedly struck by similarities between the twins. Both laughed uncontrollably, erupting into peals of giggles with little provocation. They played pranks on the staff and on each other. Both had grey hair and had dyed it an unusual shade of auburn. Both had fallen down the stairs as children and broken their ankles. Both scored similarly. Both had, albeit clumsily, taken ballroom lessons and met their husbands through ballroom lessons. Again, we are talking about twins who have been separated at birth. Secondly, 2 men, both renamed Jim after adoption, had been separated from each other 37 days after the birth. Both had struggled through school. Both drove Chevrolets, and both smoked the same brand of cigarettes. Both loved sports, especially car racing, and both hated baseball. Both Jims married women named Linda. Both had dogs named Toy, and both had sons named James Allen. Even though they had different spellings, both suffered migraines and lasted half a day. I have 2 more examples. Shall I give?
No, that's fine. I got it. So the idea, what happens is a lot of initial human genetic studies were done on monozygotic twins and dizygotic twins. Monozygotic means the same egg has split into 2, so the genetic material is more or less same.
That is identical twins?
Identical twins. Dizygotic are 2 eggs released, fertilized by 2 different things, but sharing the same in utero environment, but are like brother, sister, sibs. They can be male, female. So there are a lot of studies on monozygotic and dizygotic twins which tell us about inheritance, and monozygotic twins reared apart because they give you a different environmental upbringing, diet, all that. And it has been, and most of the inheritance percentages which we talk about today, like diabetes is a heritable disease. It's not single gene, multiple genes are playing a role. Food is playing a role, lifestyle is playing a role. But it is seen, we say that the heritability of diabetes is 40%. These percentages of schizophrenia is 60 to 70%, or bipolar is 48%. How do you say it? We say it on twin studies read apart. So it's the percentage of heritability. That means that is the contribution of the genetics. Remaining can be the circumstances,
That's what my main question, or
some of these things may be coincident.
could be. Yes, of course there might be, because out of thousands of cases we are taking only 10.
So that's why. However, there is a percentage of heritability, like migraine. I would take it as heritable. Preference of certain preferences, like getting addiction to a particular type of smoking. That because nicotine percentage in addiction is genetic. Likes and dislikes. Many of them are, see, you both are married, you don't know each other from married. But certain likes and dislikes, while living together, you learn. So that's why percentage in the whole of inheritance, I can get. But other things can coincidentally.
Because what I'm trying to ask here is, of course, the phenotypes are decided by genes, right? In some cases, some accident might change it. But is our behavior and our character and our personality...
A percentage of your characteristics are also genetic. That's why they have found that chromosome XYY has a lot of antisocial behavior. People who have been institutionalized, that is, are in jails or in mental asylums, psychosocial behavior, psychopathic behavior, they have a genetic basis. But we try to underplay it, because what will happen is then you'll blame everything on your genes.
Genetic discrimination would start.
That's the reason. So there is little bit of psychosocial behavior which is controlled by your genes, but so I don't know the 30% or 50% or what percent, because I have not done so much of neurobehavioral psychology. But I'm hoping my son would do it, because he's doing neurosciences. So let's see if he...
But it would be very interesting if that is the case. I would be fascinated, because then it makes humans just, pawns who are just playing out what they're supposed to. As it is, the geography is, for us, is decided. Wherever we are born, we live in that society. But even a character, because people put a lot of importance on what they think. Like, this is me, this is my identity. And if that is blurred because of genetic... it is going to be a riot.
Yeah. So I think a percentage of it will be genetic.
There's no denying that.
No denying that. Because we know autistic behavior, where you are emotionally detached, is genetic. Correct? That loss of emotional contact, that is genetic. Overtly friendly behavior is genetic. Attention deficient is genetic. How much attention you can...
like those are very extreme examples. So there might be some subtle examples also.
Compulsive disorders, OCDs are genetic.
These are all behavioral.
And these are all behavioral. So how much of this is, because these are complex traits, single gene doesn't control it. There are multiple genes controlling it. So what is the contribution of each one to make you behave like that?
I find it mind-blowing, honestly. Okay, so ma'am, you were talking about targeted gene therapy in college, in the talk that you gave. Can you briefly tell us what it is about?
Yeah. When we talk about, most people believe that if there is a gene change, if you replace that gene with a normal gene, then everything should be fine. So, the concept is very simple, that supposing we are talking about thalassemia. Thalassemia is a single gene, which is hemoglobin you are looking at, and you are looking at a single change in that gene. Now, what's difficult in replacing it? Because both the genes are mutated in this, if we put one normal, it should be fine. But what happens is, we have the DNA, we have the ability to make a gene which can be replaced, we have the ability to integrate this gene. But what happens is, the current method is, we put multiple copies of that gene. Because we are not sure that if we take one and put it, it will get inserted. So we are putting multiple copies. When we are putting multiple copies and it gets integrated, if it gets integrated at some other sites, then it may not express correctly. We are worried that if it gets integrated, disrupting some other gene, causing problem, or if it gets integrated and has no function because it's not close to the regulator. And that is why we started using vectors to deliver it to DNA. Now the question is, are the vectors going to get integrated and cause more problems? Because the vectors which we are putting in are mostly viral vectors which are getting integrated. So all this was being streamlined. The other thing is, most of these genes express in specific tissues. So like if you are taking muscular dystrophy, we want that gene dystrophin to express in the muscle. Now by the time we have diagnosed the disease and the baby is born, the muscle structures form. Now which muscle will you put it? Cardiac muscle, leg muscle, hand muscle. So the delivery becomes difficult. So it is very easy to do it for blood disorders, gene therapy, where we replace a gene, but it becomes difficult for organ-specific diseases. Like if I am doing spinocerebellar ataxia, where the brain, neurological things where we need to replace a gene.
It's a zero error margin.
Yes. So the tissue specificity of gene expression makes gene therapy difficult. The same genes are there in your stomach. The same genes are there in your skin. But all the genes are not expressing. We don't want gastric juices coming out of our skin. So it's tissue specific expression. That is the difficult part. Delivering synthesizing a gene which is normal, delivering it to the cells, all that has worked out very nicely in vitro. Now in vivo, these are the difficulties, and that is why we are having issues with gene therapy. But having said that, there are 140 gene therapy trials going on at different clinical stages. Clinical 1 trial, 2 trial. So it is going to happen. The only thing is,
I was also wondering about the slides that you showed in the college. You were talking more about the tests that you do to recognize on a macro to micro level. So a little about that.
The types of tests.
The types of tests.
Yeah. See, the DNA which we have is packaged with proteins to make chromosomes. So these are the chromosomes are the larger bits which we can study. So that's the first test which was done, which comes under cytogenetics, where we look at a specific number of chromosomes which are species specific. For humans have 46 chromosomes which are in 23 pairs, and it varies in the males as XY and females as 2Xs. So the number of chromosomes, if it varies, you get diseases. You get Down syndrome or Klinefelter. So studying of chromosomes or cytogenetics or karyotyping is one test. Now, numbers are easy, but if there are structural changes, again that's easy, which you can visualize under a microscope. But if this structural changes, that is, bits of chromosomes are lost or duplicated or translocated, they change place. Genetics is easy because all our words are English language based. So translocation, deletion, duplication, everything. So it's Oxford University Dictionary. So if these are large enough, again we can visualize under the microscope. But if they are smaller than that, which cannot be visualized by the magnification of a microscope, then we develop techniques like FISH, which is fluorescent in situ hybridization. What we do is, we know that chromosome 13, 15, 5, small regions in this, if they are deleted, then you get a particular disorder. So what we have done is, we have artificially synthesized that region, attached a color to it, and on the chromosomes, you put this so that it hybridizes or doesn't have. If that piece is not there, it will not hybridize. As a result, you will not get the signal. So this technology has been advanced. further down, if something smaller than that comes, so retinoblastoma, Prader-Willi syndrome, Angelman, all that, where small bits are missing, we do by fish. If it's smaller than that, but it's still larger than a gene, then we do microarray. In microarray, the whole chromosome, which we can look at, identify, shows up to by fish and this, up to 2000 regions of it we can do. By microarray, we can do it by 350,000 or 750,000. So you take smaller bits of it and analyze. So that's microarray, and then that became possible because of the computer technology. We cannot visualize it, we don't see it, we just interpret its presence or absence based on regions which are missing by signals or chips which are interpreted by a computer program. Then we come to the other part where we look at the genes. Gene present or absent. The simplest example is a child with ambiguous genitalia. It's a male or female you want. So the SRY gene. So SRY gene, if it's not there, it has to be female. So the ambiguity you are seeing may be because of extra hormone. And that can be rectified by reducing the hormone or by surgically removing that. Because in utero sometimes, like if there's a twin, female and male twin, sometimes the male testosterone goes through the placenta to that, and the other female twin is also born like a male with a little bit of enlarged clitoris which looks like a penis. But that can be rectified by reducing that. So that sort of thing. If SRY is not there, we can safely say it's a female. It may be hormonal, whatever, we can rectify that. So in doing the test, we just want to know the presence or absence of SRY. So we design primers, that comes the molecular part. That's all the advancements which have come after the technology of polymerase chain reaction came up. PCR, which is the most common tool in genetic diagnosis. So PCR, we can take a small amount of sample from the patient, isolate the DNA and do the primers for SRY. If it's not there, not there. So simple. Then there are certain genes which are there but altered, or so-called normal word is mutated. Now the terminology is changing, we don't call it mutated anymore. Now I think mutations are all in Pokemon and all, X-Men and all. But we say sequence variation, DNA sequence variation, which is pathogenic, means disease causing. Likely pathogenic, may be disease causing or may be associated with a disease. Variant of unknown significance means we do not know its significance as yet. Then there are benign, which are like sometimes, like I told you the molecular biology, that ATC is there and ATT is there. Both may be coding for the same amino acid. So then it doesn't change the sequence. So then we call it a silent mutation, we used to call it. Now we call it a benign. So now we call it a sequence variant, which is not. So the word mutation we are giving up on because mutation was always associated with a negative connotation. But a sequence change doesn't have to be negative. It can be an advantage. Supposing I've got a mutation which makes me muscularly strong, then it's not negative. I can play basketball, I can run, so it's an advantage.
That was one of very surprising thing that I realized, that yes, mutations and variations in an organism does not always equate to being disease ridden, or even if they are diseased in some physical setting, it might be to their benefit.
Yeah, the sickle cell anemia is one of the sickle cell anemia is one of the best examples. Not only that, the 50 gene hypothesis for diabetes in India.
I'm not aware of that one.
That is, Indian population over years evolved to live with very little carbohydrate. We did not need much carbohydrate and our body functioned fantastically without it. Now we have got plenty. That's why our sugar levels are going high and the insulin production is not sufficient to take care of the carbohydrate we are taking in. So that was an advantage at that time. Now it has become a disadvantage.
Why is it that we require less carbohydrates?
Our metabolism, we evolved to that level. That was sufficient for us. Like people do know, they eat less and their body requires that amount. So over the years we did not have that much of carbohydrate available. Unless rice cultivation, wheat cultivation is all. China se rice aaya, and wheat came from Brazil, tomatoes came from Mexico and all. So leaves we thrived on. Leaves don't have carbohydrate. हमको पत्ते we knew. That's why we have so many sabzis. They don't have. उनका एक ही lettuce रहता, बड़ी मुश्किल से spinach आया.
They even have broccoli here. It's not even leafy.
So broccoli is anyway synthesized. It's not.
It's not. All four, na? Broccoli, cauliflower. But this is it's a worrying thing seeing all the junk food trends that are coming in now that we're importing.
Yeah, so maybe we have to think that we also evolving genetically.
But does it happen at such a quick rate?
It doesn't, but it will happen eventually. So we have to think
eventually.
timeline. So we are at that phase.
And ma'am, can you also explain why sickle cell?
Yeah. Sickle cell anemia is a mutation, a single gene, single nucleotide change in the hemoglobin gene, which alters the structure of the red blood cells. Our red blood cells are spherical, and this alters it to make it into a sickle form. Why did this adaptation help? Because malaria parasite, part of its life cycle occurs in the red blood cell. So when the red blood cell is no longer round, it cannot function. So its life cycle is disrupted, and as a result, that person is protected from malaria. And because malaria was endemic in Africa and India, people with sickle cell disease
had a greater chance of
advantage, and they survived. And they survived only in heterozygotes. That means if they had one normal gene and one sickle gene. If they had both sickle, they died because the hemoglobin was not right. If they had both normal, they did not survive because they succumbed to malaria. So heterozygotes survive. When heterozygotes marry, then again you get percentage, 25% of homozygotes, 25% of this, and 50% of heterozygotes.
Yeah, so the mutations that are still surviving are not there just for no reason.
Yes.
There has to be some reason why evolution has taken it into. So, where do you think technologies like CRISPR and Cas9 fit into, let's say, of course, targeted gene therapy? And what is its situation in India right now?
See, CRISPR-Cas9 is just fantastic because, like I was telling you, gene therapy, we were putting in multiple for it to get integrated into DNA. Now, with this, we can integrate it exactly in that region where there is a defect. So, now we will not affect anything else. We will just go and change it. It has worked successfully in 2 cases internationally, one in Canada, one in US, both for leukemia, blood cancer. Because you have to understand, blood is the easiest thing which we can take out and put in without affecting anything else. So CRISPR-Cas9 has worked. One thing we have to appreciate is the FDA gave approval. The other thing you have to appreciate is that the family persisted. The family and the advocacy groups, they pushed this. And that is where I feel India lags behind. India doesn't have many advocacy. Now it's happening. Rare disease groups, both which are functional in India, are run by parents who have affected children. Prasanna Shori work in rare disease in Karnataka, and another one who's promoting Vikas Bhatia for newborn screening. Both have suffered because Vikas lost 2 of his children to newborn errors of metabolism, and Prasanna's daughter is still alive with a metabolic disease. She's wheelchair bound, she's mentally and physically affected. And that's why I said the driving force sometimes is personal. Advocacy groups now, we are asking genetic counselors to take up and promote advocacy groups, and we have started one in Kaminini. Yashodhara was my student who started it, and she started it with 200 cases which we have, and tried in which is included thalassemia, although thalassemia no longer comes in a rare disease, but we still call it rare because clinicians don't identify it. And our babies are dying before they get diagnosed.
So is there a different protocol for rare diseases? And
No, the unfortunate part is if somebody has a rare disease, the clinician loses interest, pharmaceutical companies lose interest, and then the child, individually it may be rare, but collectively there are a big number. We have more than 30 million children with rare diseases in India alone. And what is more important is that the same parents can have a similar child if it's not diagnosed properly. And that is failure of the medical system and the healthcare system. If 2 children with thalassemia or 2 children with mucopolysaccharidosis, Gaucher's, it's terrible. That means nobody is ever killed. I've got from LV Prasad families with 2 children with retinoblastoma, which is an eye cancer, bilateral eye cancer. If one was there and coming to you, you should have told that this is genetic. Fine, you didn't have a genetic unit, but you knew the disorder was genetic. They could have gone somewhere to get genetic information. So prenatal testing could have been done. We could have shown that this is next child also is affected. Can you imagine how devastating it is for a parent to have a child with, and then 2 children like that? So CRISPR-Cas has come in our labs. Institutes are doing it, checking it out. But our drug controller for India treats everything in one basket. Even diagnostic tests are treated like a medicine. So it is sad, but maybe that also. I say, nakhal karne bhi akhal chahiye. Correct? So you're copying certain things from the West. So do it at least fast and do it quickly. That okay, this is important for our country. Let's focus on it, work on it.
First of all, I've seen whenever some revolutionary things, or not even revolutionary, there is a technological advancement that India wants to adapt. First of all, there is a lot of retaliation. Yes. When it finally comes, it has already progressed in the West, and then they adopt it, and they adopt it in a manner where it is not applied everywhere. People are not educated about it.
But then there's some advantages to being slow.
Yeah.
One was the thalidomide story. You don't know, in the 1960s. Early 1960, a drug, thalidomide, was developed, and that was for suppressing nausea and vomiting, and it was considered a great boon for pregnant women who suffered with this morning sickness. And then the children born, this was in Australia, Europe, it was all developed countries used it right and center. 9 to 10 months later, there was an epidemic of children born without limbs. The limbs were not formed, trunk, and those were the days there was no ultrasound. So they couldn't be detected. They were born. And they thought it was some infection, virus, all that. And then they realized thalidomide was the drug which interfered with the development of the limbs. So luckily we had no case in India because the drug was not introduced. We were slow. Otherwise I am the 60-61 born person. I could have been in that category. Yes. So whenever I see, nowadays sometimes I see when I'm traveling internationally, thalidomide babies who are now adults like me, on wheelchairs and some of them who have survived. And I know that, see, this is the advantage of being slow. Yeah. So about 20 years behind.
So I was also wondering whether anywhere in the world, the experiments and the testing with CRISPR, is it being done on germline cells?
See, CRISPR, one was done by the Korean meeting, and he is now being put behind bars. He did by CRISPR-Cas CC3 CCR5 gene to parents who had HIV. HIV is transmitted vertically also, but CCR5 gene is the one which protects from getting the AIDS virus replicate. So what they did was in the in vitro germline, they changed that gene for the child to put a resistant gene so that they are protected from AIDS. And he claims that he was successful, but he was put behind bars because he didn't take all the necessary approvals. So they can do it. So now it's not being done, but it's possible.
But there are regulations right now that are not allowing it. I read the Jesse Gelsinger case.
So the ethical concerns are there about, they make it sound like designer babies. They say, is it ethical to have a designer baby? But technically that is not a designer baby.
It is not enhancement. It is
more of a protection. Protective. So it's not, in my opinion, what he did was not wrong. The way he did it was wrong. He did not go through the approvals. But if he had done the same thing 20 years back, it should have been fine. Now all these ethics committees and many people, if you see the whole ethical thing, and the human genetics carries the baggage of the Holocaust, eugenics. So we tread very carefully around that. Whereas in other specialities of medicine, they are much more stronger. They push into it.
Especially China, like it has no holds barred going ahead with it. Do you think the matter of genetic manipulation will become a central area of discussion in the future?
It will, at least for disease, like Duchenne muscular, and it should, or thalassemia. As of now, we are doing bone marrow transplants. The cost of it, the procedure, all of that is success. Because if there is a graft versus host, this disease and so on, or you have to give immunosuppression lifelong, all that will be changed because you'll just be inserting one gene. The rest all you are not putting somebody else's. So it has to come, it will come. And the question is when. For the common, once we establish the incidence and indicate to the policymakers that these are no longer rare diseases and the numbers which are suffering from it, the disease current treatment, the cost of it, and the problems associated with it, then it has to become. Leukemias and cancers, people are more ready to try it because they are adults. We know what the treatment is, we know what the prognosis is, and we know what our limitations are. So, more acceptance is there for those disorders.
And I think not just treating patients who have already grown up with the disease. Once it becomes evident and apparent and spread throughout the world, in vitro checking and...
In vitro testing, like in utero, you mean?
Yes.
In utero testing we do a lot. Even now, we do for chromosomal disorders, we do for special genetic, if we know families which have a particular gene mutation or pathogenic variant, we do the amniocentesis, and we are doing it now, not us, various centers in India are doing it, not just us, we at Kaminini do it, and that's what I said. If a second child has the same disorder, that's our failure. There's a failure of the health system. Of course, religious and cultural issues come into it, like certain people who do not believe in termination, they may not want to do prenatal testing, but that comes with genetic counseling. We do the pre-test counseling, and in the pre-test counseling we inform that this condition as of now cannot be treated in utero, so termination is the only option if we find a problem. So if they do not agree to it because of religious reasons or cultural reasons, like if they are Muslims or Catholic, then there's no point in doing the test at that. You can do it when the child is born. So the option is always given to the parents. Legally in India, up to 20 weeks, we are permitted to do it. But before 20 weeks, that again is difficulty, because France and Germany, what they have done is they permit till any stage of pregnancy, provided we know that the condition is not treatable with. So those changes, here what happens, for every case you have to go to the Supreme Court. Who can afford it? So that's why what happens, illegal things happen. Illegal terminations, which are not right.
And knowing just the time span that it takes for a case.
So the Indian scenario, the major lacuna I think which we have is the lack of genetic counselors who can bridge this gap, who can talk to clinicians, who can talk to patients, who can talk to families, convey them and keep up with the recent advances, availability of tests. Before we used to send for BRCA1, BRCA2 also, we used to send to Centogene or USA, Myriad, and the cost was 80,000. Now it is 15,000. So the test cost is coming because we are doing tests in India. There are 6 or 7 companies in India who are doing it. Once a sequence variation is known, to confirm it and all, we do it in Kamini itself. The whole sequence. NGS we don't have. NGS is the platform for doing next generation sequencing. That's the advanced sequencing thing, where you can take the blood DNA or saliva DNA, amplify it by PCR and do parallel sequencing of the total DNA. So the cost has come down, time has come down. Human Genome Project to do the whole DNA sequencing took years. Now you do it overnight. You put it in the machine and
even companies like 23andMe, you can just send your saliva and they...
You can send it to us also. We can. Yes, we can do it, but we don't do. They do SNP based. So they do, like for breast cancer, they are looking at only common targeted mutations. So if a positive thing comes, we can indicate that there is a problem. But if it comes negative, we are not able to tell you by 23andMe. So the whole sequencing is better.
And 23andMe is also mostly aimed at ancestry and not disease.
Now it is marketing ancestry because that's the selling point.
Okay, so it's a very personal question to you. Who do you think, in your opinion, has made the biggest contribution to the world of genetics?
See, the Human Genome Project has done a lot for it. So that Khurana did it by getting that money from the US government. Francis Collins is doing a lot, being the director of NIH, by translating it. Then there's the Human Genome Foundation and the NHS is doing it. Anna Middleton is doing a lot for the ethics of it.
Wow, that is also a very
important
central part of...
She's doing a lot through genomic and then Wellcome Trust and all that. Craig Venter showed that even individuals can contribute. Although he became a bit selfish and self-centered, but like they say, now there are very few individuals who can contribute because consortiums are contributing. Consortium of neurological disorders, psychiatric disorders, which you are interested in, behavioral psychiatric disorders, consortiums of Australia, who is looking at totally different aspect, the Octarian thing. So they are mostly consortiums now. Another person who I think has helped the Indian genetic genomic education is Dr. Devendra Kumar. He's from India, lived all his life in the UK, and at a senior level he wants to contribute back. So he brings in expertise of genetic education in different fields like ophthalmology ones, neurology ones, every 2 years to India. He motivates UK's scientists to come to India and takes them around to different academies, different institutes in India. So I think more and more we are moving towards single people. There are multiple people, multiple groups who take up. Of course, we also have now Dr. S.S. Agarwal's baton has been taken under Shobha Phatke. She's running that DM Genetics program. And her students, they have made one society for medical genetics. In NIMS, they are there. In Hyderabad, NIMS, Ashwin Dalal, Shagun Agarwal, Pragyan are there. So there are various groups who are coming up. So that's why I said just naming one would not be fair. It's the whole, yeah. And also my students, because they did genetic counseling more, and they are bridging gaps with clinicians. So oncologists are coming forward. Rajiv Sareen in Tata Memorial, when he was a doctor, he was the one who set up a genetics unit. So there are lots of people who we need to recognize. Yes. So I don't know. I'm maybe getting old too and forgetting names, but
already you're saying that your students are bridging gaps.
Yes, so let's hope they remember to acknowledge the right people. Because that's one thing which we see that youngsters forget, because in India we don't teach them history properly.
Or the way history should be absorbed. And semi-last question. To wrap up the conversation, let me end it on a sort of a fictional question. How much do you think is there a possibility of humans enhancing themselves with cybernetic technology or through genetic engineering? So, for example, rather than for getting better eyesight, rather than getting a better gene, someone installs enhanceable zooms in their eye lenses. So, will it be a marriage of both, or which one do you think is leading the frontier right now, and which one would be in the future?
See, relying on external devices, we've always done. Using of glasses, using of hearing aids, pacemaker, all kinds of things. But there's always a reluctance of putting something inside, which is non-biological, because of the reaction to it, or we know that it is different. Implants for teeth. That's been going on for years and years. But correcting the biology is the novel one. And people feel that, okay, then once if you do that small correction, there's no, you don't have to keep changing glasses, and it's yourself. That's why there's so much interest in stem cell therapy. Now the regulations and also the coming of the 3D era in biology, that will be the major game changer. Because on one side, like if you're keeping on giving insulin injections, and instead you can take in the pancreatic beta cell, which are going to produce insulin, everybody will opt for that. Where there's no major intervention, but you're just injecting something like an IV line and putting cells which go home there and start building. Same with most people now are accepting knee replacement, hip replacement.
But if I try to defend the side of cybernetics, in the case of where someone or an individual wants more of an extreme version of the same thing, whatever we do, we cannot make bones as strong as, let's say, steel. But if somebody wants an enhancement,
then you would go for it.
Yeah, so I'm wondering, will that be a case?
Now, the question is, how different do you want to be from others? That's why all these Avenger movies are so fascinating to people. And what I find funny in them is that they have all these powers and finally they end up fist fighting.
Yeah. One simple kinetic manipulation can change the whole stage.
Why are they doing fist fighting or killing each other?
But surveying the social medias, it is evident that everybody wants a unique identity. And whenever a new option arises, they just grab onto it.
Height, enhancement of height, getting that extra push for running, looks. I'd gone to Korea for a meeting, Seoul, and in the plane I saw there were people with bandages and all. They do this.
I only read about it. Is it
really
evident?
Yeah, they do it. All our actresses do it, so anyway, now individuals do it. They are obsessed with their skin. I've never seen so many shops there for their skin. Snail mask for your skin, this mask for your skin. I thought I should buy some and try it, but I didn't. I'm not. Some reaction I'll get a little worse than what I have.
Okay, and one last question, which is a very, it's a topic of, it's a central interest right now in the world going on. What would you say genetic engineering and genetic studies, how do they comment on gender identities as of now?
Yeah, as of now, the last I read, because this is a question which my husband has often asked me, because when you talk, when he wants to talk to conservative people or liberal people, we stayed a large chunk of our life in New Zealand, which was very liberal, and even in the 90s, we never asked anybody about their spouse, we always said, bring your partner along. So we never knew what the partner would be, the same sex, different sex, all that. Genetically, we were not able to identify differences even in the most recent literature. The thing is, if you see mating types, there have always been two mating types. As in evolution, the first mating types you see in Paramecia. When they are for adverse conditions, they have reproductive. Otherwise, they have asexual reproduction. But for adverse they have. And then it becomes two mating types. Then came the fungus. That also has plus mating and type and minus mating type. And
so again it is binary.
So it's binary. So where has this come from? So is it a preference more got because you have needs other than the sexual need of reproduction? Because reproduction, we are now looking for IVF, for this and that, for surrogacy and things like that. So now I think people are trying to re-educate you. They talk about bonding and pregnancy and to make you feel that, okay, get back to this. Don't go have a baby through surrogacy. We are trying to do all that. Or breastfeeding. Why are we promoting all these things? You can have a breast bank and feed them. Correct? The question still remains that is there a necessity of more than two mating types, or is it just your sociological, psychological need where you feel more comfortable with one, the same sex, more than the other, and where do you come to the third sex? Because the third sex is not at all there for procreation. So then where does it fit in? So is it an aberration? Both a psychological, hormonal, physical, and what is the genetic variation in that has not been identified as yet, and I'm sure they are getting into it and working towards it to understand.
Because even I was in the belief that binary does the job and that is where the definition should stay. But when I read about XXY, XYY,
they are aberrations. Then that's why I'm saying XXYs are males because the SRY is there. We've just XX, the chromosomes are 2 X's, but the SRY gene by mistake, by translocation has come on the X chromosome or any of the other autosomes. So SRY is there. In XY females, the SRY is deleted or suppressed.
So there is one suggestion by people that maybe we assign them their genetic gender at least till 18 years of age and then they can decide for themselves.
Yeah. So that is what. So what do we call gender? Chromosomal gender, genic gender. One is chromosome XX, XY. Genic gender is SRY, present or absent. Or downstream thing where there is one more syndrome where it is XX but no uterus formation because then the uterine genes are not upregulated or deleted. So then they are not functional females. Functional means, but if you phenotypically they are female. So now the question comes of either we have to identify the need of it, that in evolution they have come up, but it's not that because they have been identified. So then it is more hormonal or psychosocial, which makes you feel. Because in childhood, many boys like dressing up like girls because it has more of ornaments. Wearing those bangles, wearing the things.
And there have been cases where parents take those indications as their decisions and they already start hormone therapy when they are little children. So I don't know what is the answer, but at least
we have to think about it as more and more such things come up into the open. When something is hidden,
it cannot be discussed.
Now, let's see. But as of now, I did read one where they have taken a particular population on these and done the genotypes, and they have not been able to pinpoint a variation. But maybe the numbers are less, or they are not looking at the right gene because we are looking at maybe only hormonal, and it may be something totally different which will be causing it. Maybe looking at androgens, testosterone, progesterone, cholesterol, or something which develops these precursors, and they may not be looking at something which is totally different and existing.
Maybe after the complete sequencing of the human genome that is going on. By complete, I mean every intron, exon,
it can, that is done, but correlating with the phenotype.
Okay. So once we know what everything does,
and
then I think a lot of questions will be answered for us.
A lot more than what we know. Yes.
I think we can end it here, ma'am.
Thank you
so much for giving us your time.
And now I have to do a little bit of my job.